my name is Edwin Kwon, and I am the Chief Financial Officer at Eutilex. I am pleased to give you a brief introduction to our company, followed by an introduction to two of our pipelines for which we are actively seeking partners. As you can see, our name comes from Latin roots, EU meaning new, TI meaning immune therapy, and lex meaning principle or method. So taken together, our name establishes what our company does, which is developing new breakthrough immunotherapeutics against incurable diseases. We are a public company in South Korea, so here is our disclaimer regarding forward-looking statements. Now, Eutilex is based on three world-class technology platforms, each of which having multiple product pipelines with blockbuster potential in oncology. This slide has a brief overview of our platforms. First, we have an antibody therapy platform in which we have developed best-in-class and first-in-class immuno-oncology antibodies with higher efficacy and new modes of action. These antibodies have already demonstrated their potential to become blockbuster candidates in the immuno-oncology market. We have EU101, a superior 4-1BB antibody, which in a moment I'll discuss further. Our EU102 is an efficacious AITR antibody and exhibits a brand new, unique mode of action. EU103 is another immuno-oncology antibody with a first-in-class mode of action. And not listed here are our bispecific antibodies and more than 10 other antibodies in our development pipeline. Next, we have Eutilex's own T-cell therapy platform, which has already shown very significant results in clinical trials. We've even seen outstanding safety results and compelling signs of efficacy in solid tumors where other cell therapies have not come close. Our product against Epstein-Barr virus-induced cancers is now going into phase 2 after a remarkably successful phase 1, and we've seen convincing data in solid tumor clinical trials which are now in several phase 1 studies. We are also developing new ways to address the heterogeneity in solid tumors with our TAST program, and we're doing that through investigator-initiated trials. And we also have a preclinical COVID-19 therapeutics program, and that was selected as a national project in Korea. Our third major platform is our superior CAR T-cell therapy platform. And in our MVR CAR T-cell therapy, we boast higher safety by not attacking healthy B cells as prior CAR T-cell therapies have done, such as the older CD19-based CAR Ts. We are also developing a CAR T-cell therapy for solid tumor, and this is another pipeline that I will discuss in more detail. Finally, we are working on a universal CAR T-cell therapy, which would be an off-the-shelf CAR T-cell therapy, allogeneic, one for everybody. With these three platform technologies, Eutilex has the pipelines in development to become a global biopharma leader in the immuno-oncology field. Here is our company founder and chairman of Eutilex, Dr. Byung-se Kwon. He's worked in immuno-oncology for over 40 years and is a giant in his field, an internationally renowned immunologist. Having produced a Nobel Prize level body of work, he received the Star Faculty Award, which put him on the short list of Nobel Prize candidates. He has been a distinguished professor at the National Cancer Center in Korea, the University of Ulsan, a tenured professor at Indiana University, and he's also researched at Yale University. Among his other achievements, he was the discoverer of 4-1BB and AITR, as well as human HVEM and DCR3. He's been cited over 17,000 times in scientific journals. As you can see on the left, Eutilex went public on the KOSDAQ at the end of 2018, and our highest market cap has been at almost $840 million US. Currently, besides our office and lab space, we have a GMP facility and an animal laboratory. Right now, we are over 100 employees strong, and 45 of whom are researchers. Eutilex has a first-class, well-rounded management team with a long list of proven achievements and an international professional background. Our team has extensive experience in regulatory affairs, commercialization, multinational companies, patent development strategy, and quality control. This slide gives some context to the technical capability of Eutilex. As you may know, T cell activation has two mechanisms, and they can be divided into the immune checkpoint regulator categories of immune checkpoint inhibitors and checkpoint activators. In 2018, the Nobel Prize for Medicine was awarded to Professors Allison and Hanjo for their discovery of cancer therapy by inhibition of negative immune regulation with antibodies. Likewise, Dr. Kwon is the foremost expert in immune checkpoint activators, having discovered 4-1BB, AITR, and HVEM, among others. Now, several treatments have already been approved under immune checkpoint inhibitors, including Opdivo and Keytruda, and these work by blocking inhibitors and allowing the T cells to fight the cancer cells. So if you think of immune checkpoint inhibitors as the brakes of the car, we at Eutilex have the expertise in immune checkpoint activators, which would be like stepping on the gas pedal in the activation of T cells. Before we move on, I'd like to give a little background on the immune system. Now, as you may know, 80% of cancer killing is done by killer T cells. They can kill cancer cells at any stage of cancer development, and they have the ability to infiltrate into solid tumors. Thus, the major workhorses in immuno-oncology are killer T cells, and so the focus is on activating killer T cells. In this slide, we can see how killer T cells are activated, which is necessary before they can kill cancer cells. Dr. Kwon, our founder, is the discoverer of the principle of killer T cell activation, and this diagram here is now very typical of those that are found in immunology textbooks today. So activation of killer T cells is a two-signal process that is dependent on the cancer antigen. Two signals are required, one of which is cancer dependent, to tightly regulate T cell activation, or else they might attack healthy cells with serious side effects. The first signal is an antigen-mediated signal. As you can see here, a cancer cell will present with an antigen specific to the cancer on its surface. The corresponding T cell has a receptor that recognizes the antigen and binds to it. However, antigen activation also requires the presence of a co-stimulator, and that's the second signal, a co-stimulation mediated signal. There are two kinds of co-stimulation factors, constitutive and inducible. The constitutive co-stimulation factor, as we see here, is not specific for the cancer antigen, but is expressed on all T cells. Therefore, early attempts to activate T cells through constitutive co-stimulation factors, such as CD28, cause serious side effects, often fatal, because all T cells were being activated, not just the cancer-specific ones. However, all platforms at Eutilex use inducible co-stimulation factors, which are very safe and effective, because the inducible co-stimulation occurs after the T cells already went through the constitutive co-stimulation and the antigen-specific activation. Now, under normal circumstances, the chances of these cells meeting one another and binding are rare because of the number of cells and the space in the body. However, we can use 4-1BB and AITR antibodies to replace this ligand activity and activate T cells that are cancer-specific killers. As I previously mentioned, Dr. Kwon is the discoverer of this activation mechanism, which is the basis of all current immunotherapy treatments. Eutilex platforms are based on his expertise in this area. Now I'd like to give a more detailed introduction to one of our pipelines, EU101, a 4-1BB targeting antibody therapy that uses a completely different framework than previous generations of 4-1BB antibodies. We are currently preparing our US FDA IND filing, which will be submitted during this calendar year. Our antibody activates the T cells and promotes them, increasing their numbers, and I'll show you in a minute the extraordinary cancer-killing effect. Here you can see that we tested the efficacy of EU101 on 4-1BB knock-in mice. In these experiments, once the tumor is established at around 100 cubic millimeters, we started treatment, and what we see is that the tumor growth is substantially inhibited. We tested at doses of 1, 5, and 10 milligrams per kilo. As you may know, a bell-shaped response is typical with efficacious treatments in knock-in mice, and here we see that typical response with our most efficacious dose at 1 milligrams per kilo. We've also examined the tumor-infiltrating lymphocytes in individual mice, and what we observed was high infiltration of cancer-killing lymphocytes into tumor tissue. In particular, you can see that in the CD45, CD3, and CD8 graphs, that with EU101 as opposed to the control groups, there are enhanced infiltrations of cancer-killing human leukocytes in the tumor sites. Additionally, in the Treg graph, we see a reduction in Treg cells, which shows an improvement in the tumor microenvironment since Treg cells inhibit cancer-killing T cells. Now, very exciting for us is that we've observed no drug-related adverse events in our experiments with EU101. The no observed adverse effect level in our GLP monkey repeat tox study is 200 milligrams per kilo, and the maximum tolerated dose was considered to be 400 milligrams per kilo. So all of our data points towards safety in man. In summary, EU101 inhibits tumor growth, induces infiltration of cancer-killing lymphocytes into tumor tissue, improves the tumor microenvironment, and has shown no drug-related adverse events in monkeys. EU101 is proving itself to be both highly efficacious and low in toxicity, all in one molecule, and we consider this to be an achievement that will have a global impact. Next, I'll introduce one of our CAR T pipelines, our GPC3 CAR T cell therapy, which addresses solid tumors, and which that has been very difficult for other cell therapies. Our target is the GPC3 protein. Importantly, anti-GPC3 has been identified as a useful tumor marker for a number of cancers, including liver cancer. And in patients with liver cancer, GPC3 is overexpressed in the cancer tissue, but in healthy persons, GPC3 is undetectable in their normal cells. This is why GPC3 is such an attractive target. Liver cancer ranks as the 4th highest number of cancer deaths in the world, so it is a significant market and it is our target indication. Here is some further information on our GPC3 CAR T. It is an autologous CAR T with an indication of liver cancer. We are using a 4th generation co-stimulatory signal domain, and we have confirmed in vitro efficacy in liver cancer. On the right, you can see the mechanism of our GPC3 CAR T cell therapy. We've developed a GPC3 CAR that secretes IL-18, which improves the tumor microenvironment, as well as improves the survival of the CAR. Here we share some of our in vitro results. On the top 2 graphs on the left, you can see that in cancer cells that express GPC3, we observed high cancer-killing activity by our GPC3 CAR T. On the bottom left graph, we observe that in GPC3 negative cells, we see very little cancer-killing activity. This goes to show the safety in our approach. On the right side, you can see that we had a very high CAR expression in our GPC3 CAR. Moving on to our in vivo data, you can see on the left side, we observe that the suppressive activity to be far greater in our GPC3 IL-18 CAR T as opposed to previous generation CARs. On the right side, you can see that we observe that our CAR stays longer in the blood than previous generation CARs. Just like in our in vitro data, on the left side, we again see that in GPC3 positive cells, we observe a high cancer-killing effect, but we see very little effect in GPC3 negative cells. Again, this goes to show the safety in our approach. On the right side, we induced liver cancer in mice to show the outstanding efficacy in orthotopic models in comparison to a control group. Finally, I'd like to share with you our timelines for the previously mentioned pipelines. First, with EU101, as I previously mentioned, we are filing our US FDA IND package before the end of this year, and we are looking for partnering opportunities as we move along with the clinical trials, which will take place in both the United States and in Korea. In parallel, we had previously licensed out the EU101 molecule for the China region only to Huahai Pharmaceuticals in China, and they have already received approval to begin clinical trials in China for EU101. So in the upcoming months, we will be able to view their clinical data even earlier than our own, and that will give us a sneak peek into what we can expect in the future. With GPC3, we expect to initiate clinical trials in Korea in the second quarter of next year, and will follow in China soon after. Significantly, the number of deaths in China for liver cancer are 10 times those as the number of deaths in the United States. We expect that we will receive expedited conditional approval after phase 2 data, and we are actively seeking partnering opportunities. I hope that you will please feel free to reach out at any time with any ideas and opportunities in which we may be able to collaborate. You can reach us generally at partnering@eutilex.com, or you can reach me personally at eekwon@eutilex.com. I deeply appreciate your time and attention to our presentation today. Thank you very much.